we have a very, very special guest tonight. For some of you, first time you've had an opportunity to meet her. For some of us older members, we've had opportunity, Ann and I, we've met several times up at the conclave in Michigan, and you've been down to the club before, and et cetera, et cetera. We won't go back and reveal how old we are, but we've done this quite a few times, and I'm very excited. And you were actually a special request by several members. They said, could you think you could get Ann Miller here? So I said, well, let me call her and see if she would be happy to help us. So she is going to put together a program, I assume is it's kind of a general thing, or are you going to lean this a little bit towards the drift list, the more simplistic entomology for us Illinois people, or?
I'm going to be very simplistic, but also go into a little detail and tease your brains a little bit, but not an awful lot. But give you a little food for thought and talk about some general entomology and yeah.
Good. Well, she's the current president of Flygirls in Michigan. She has been a force in the fly fishing industry as long as I've been involved in it, and is also a very good friend of a dear friend of ours, Tim Scott, at the Tyathon. And they have been a significant contributor and involved, I guess, on a grassroots level with them for a very long time. And it's a very important program to us as well. So, Ann, with no further ado, the floor is yours.
Okay,
And,
let me
mute their mics, please, and then we'll let her get started.
Okay, so let me let's see if I can share my screen. Yep, there we go. Everybody able to see that okay?
Yep.
Yep. Yeah.
Okay, now I'm going to do a couple different views. If I leave it like that, can you still see my face versus this? You can still see me?
Yeah.
Okay, good. All right, let's go with that. My program tonight is virtually buggy. And I want to, I mean, we're an informal group. It's fun to be back to talk to all of you. And I want you to feel free, like if a question pops into your head, please ask, you know, probably if you're thinking it, someone else is thinking it. And, you know, as we all know, the only dumb question is the question that's not asked. So feel free, we'll just kind of be informal here, and I'll get started here. So this is my oldest daughter, and I think when we're kids, we have a natural affinity for bugs until we get stung or bit or something unpleasant, and then, you know, we don't like insects thereafter until we get into fly fishing, and then we're all about bugs. And I became just really, I always loved bugs, and they became a big part of my study in college as an aquatic biologist, and then really in graduate school, I became very interested in the study of aquatic insects, and became involved with what was then the Federation of Fly Fishers, and of course Trout Unlimited, in teaching fly fishing intro classes. And since I was the aquatic biologist of the group, I always taught the entomology section, and I was very frustrated in not having any good resources, and any handouts I could find, I had to, you know, copy and paste from a million different books. And getting into fly fishing, I remember reading all the different types of flies and the insect it would imitate, and then you'd go back and forth and buy all the fly fishing, all the fly tying books, and try to match those up. And I just thought, you know, it'd be really nice if there was a book that had the bug on one side and the flies on the other side. And so that is what I set out to write. And um, I got started on that. It took me about 3.5 years to write it. I learned that collecting the immature stage was relatively easy, not terribly challenging to photograph. But when you got the adult insect back into your photo studio, a lot of times the wings would be damaged, or they might be completely dried out, desiccated. They might be just downright dead, and there's no way you can stage a dead insect and make it look good and put it in a publication. And I have seen publications where insects were glued to a stick or something, and then the picture shown. And I didn't want to do that, certainly. And I also didn't want to try to use Photoshop to replicate missing body parts. So I 6 months into this in writing project and research, I had a little bit of a panic moment because I wasn't sure how I was going to try to, find 50 some different taxon and photograph the adults. And so I had a friend in graduate school who had some success with artificial streams. And I called him on the phone, and he talked to me for like 3 minutes. And I was like, oh, he gave me some ideas. And so I set up a number of artificial streams in my garage and basement as I was working on this project. And what you're looking at here literally are eaves troughs and a recirculating pump and there's a little bit of a slope here to provide current, and then recirculating stream water, which I would bring by the carboys. And I would have frozen milk jugs in here, and I would keep these, at temperature. And then I would inoculate, I would put river rock and wood, bits of twigs and various substrate in my little stream. And then I would go out and collect nymphs of several taxon and put them in the stream right before they were due to hatch, right in the last molt. And it turned out to be really successful. And as you look at this very, primitive little stream, I've got just, a little mesh material over there. You can see insects hatching out. And I was able to very easily just reach in there and extract the adults. All the body parts were intact. I could, rush in then and photograph the insect. I could collect male and female, and it worked really, really well. And I was able to get very, very nice photographs as a result. And so at any one time, I would have many containers filled with various taxon, and that I would put into a number of these different streams. And it was very labor intensive, of course, keeping all these streams maintained, bringing all the water in, bringing the insects in, but it worked really well. And I still spent a lot of time on stream collecting the nymphs and the larvae and studying the insect behavior, but getting from the stream back home to photograph them, was no longer an issue. So my book is Hatch Guide for Upper Midwest Streams, and I'll put my email up at the end of the session. And those of you that are interested, I am going to. I know a lot of you in the club have my book. It is now out of print, and the publisher, I think has had some issues, which I won't go into, but they've let a lot of their books go, and I decided just to self-publish it, and I'll have that out later this year. I am going to add a section on terrestrial insects, so there will be some new insects and patterns in there on our various terrestrials of the Midwest. So, okay, so that's just a little background on some of the research and fun stuff that I did in writing it. You guys being a tying club might have some fly boxes like this, but all of us, the more you get into this sport, the more flies you have, and organization and choice becomes a challenge. And so often we find ourselves on a river trying to decide what fly do I put on and why. And, sometimes for some people, it might seem like guesswork. And what I want to talk about tonight is let's put a little more of the science into it to maybe help you choose a fly in various scenarios and why you would do that. And some of it will seem obvious, and some of it may be a little bit novel for you tonight. So as you look at these next series of slides, look at how this river changes. Imagine how the current is different, how the light through the water column might change, the depth, the bottom substrate. Here is a section of gravel, and if actually you look close there, you can see a lot of little cased caddis hanging on to the bits of gravel and cobble here. And certainly here in the Midwest, especially in Michigan, we have lots of sections of water that are tree covered, and you can imagine a lot of down trees and leaves and things like that in the water. And so if you have my book and you look at any one of these sections of the river, my question to you is, will you find all of those different insects in any one section of the river? And of course, the answer is no. It's no more like if you had a bird book and you looked out your window and expected to see all those birds out there. There are a lot of different variables that determine why something lives where it does. So let's look at some of that. Let's look at some variables that determine why an insect hatches where it does. So first off, important, is geography. And this might seem a little bit, obvious. Like if you go out west, there are different hatches there than if you're here in the Midwest, or, say if you went out into the east. And many of those hatches are similar, but specifically species-wise, we have differences wherever you go and around the world, and of course in this country. Second up, a variable that's probably a little bit obvious, you might not know the word, but phenology is a fancy word that just means the natural order of things. And I like to equate this to, for example, the flowers that bloom in your garden, So we start off with crocuses, and then come daffodils, and then come tulips, and then come iris, and then lilies. And that order is the same every single year. And the bloom dates can move a little bit earlier or a little bit later, but that order will always be the same. And that's great because the same is true with insect hatches. And this works in our favor. So we can go to, a section of water in Michigan or Wisconsin, and know that every year, right about now, there should be Hendrickson's hatching within a certain, few week window. And that's very helpful. Now, another variable that I think many anglers have overlooked is habitat. And as you saw those different slides, you could see that some of the river may have been completely comprised of gravel, and some of that river, like this slide shows, there's probably very little river or very little gravel in there. It's more decaying wood and leaves, maybe a little bit of gravel. And so a lot of variability there. And in each of those different kinds of habitats provides different food. And so all of these different insects feed differently. And so they have specific habitat. And in my hatch guide, I try to tell you, if you're looking for this particular insect hatch, you got to have this kind of habitat. You have to either have gravel, or you have to have submerged plants, or maybe, large cobble, maybe tailwaters. So, and then lastly, an important variable is time of day, which I think as fly anglers, we're definitely tuned into. We know that certain hatches happen midday early in the season, whereas later in the year, maybe those hatches get moved to late at night, or maybe very early in the morning. So all of these are important variables when you decide what insect hatch you're pursuing, and when you should be looking for that hatch. Okay, so many of you are familiar with the mayfly life cycle, and maybe some of you are not familiar. So I want to review that. And when we go to school, we learn one life cycle and we learn that of the monarch butterfly. And we know that we learn the big fancy word, complete metamorphosis, with, egg to caterpillar to chrysalis to butterfly. So a big radical change. But many insects have what's called an incomplete metamorphosis. And so if you think of something like a grasshopper or a cricket, and you think, okay, well, what does a baby grasshopper look like? Or what does a baby cricket look like? And there are insects that don't really have a big radical change as they grow and develop. So a baby grasshopper looks pretty much like an adult grasshopper, except it's a lot smaller. And really in the last mold, they get wings, and so we term that slow change an incomplete metamorphosis. And that's true with mayflies. And really, when you look at the nymph and you look at the adult, really all we've done is put wings on it, and then they really look fairly similar from start to finish. Of course, not at the very beginning, but. So if you consider looking on the far left of this life cycle chart, the female will lay her eggs, and then for everything I say tonight, there will be, of course, an exception. I'm just going to speak in generalities. So the eggs will grow and develop over the course of a year, and all of those eggs are the same age. So as they get to be about 1 year old, they all become active and start to emerge at the same time. And this sort of synchronous emergence pretty much ensures that some adults will make it to grow and reproduce for the next life cycle stage. So that's what's cool about fly fishing, is we sort of live for these mass emergences, not unlike the cicadas coming up, I guess. But so those nymphs will start to emerge. They'll swim to the surface, and right along the thorax region, the wings will, the thorax will split, the skin will split out, and the wings will pop out. And then we have a winged adult, which we call a dun. There are different scientific names for it. We won't get into that now. And then the duns they'll fly away, and then they sort of rest for a day or two, and then they molt one more time. And this happens away from the river, so we don't really try to imitate that. We often don't even see it. But then when it molts, we call that reproductive stage the spinner stage. And then those spinners they'll mass along the river, over the river. The males will come first, and you'll see clouds of them. You'll be sitting on the bank waiting, and you'll see clouds of males start to gather, and they'll oftentimes they'll swim, they'll fly up and down or side to side, depending on the species. And then they're emitting pheromones and waiting for females. And then if all is perfect, the females will come and they'll mate. And then the females will come down and lay their eggs. And so then the whole life cycle is complete. So as a fly angler, we can imitate each of these different life cycle stages with a fly and successfully encounter a fish. So lots of different options here. So just a little bit on identifying a mayfly nymph. They're discernible from other taxon by having 3 tails and their gills on their abdomen. A mayfly dun has 2 pair of wings, which it holds up over its body. So it sort of looks like a little sailboat. As an adult, they can have, remember the nymph had 3 tails. The wing stages sometimes have 3, sometimes have 2. And they don't have any mouth parts, so they don't feed as adults. And the whole name, the order name is Ephemeroptera, which means short-lived. And they only live upwards of, several days. They're entirely dependent on the amount of energy stored in their bodies to make it through the dun and spinner stage. And they tend to be very dull in color. So if I don't know what your computer screen looks like, but if you look at the fringe, you can see a little fringe on the wing here. And those are actually tiny little hairs. And if you looked at this with a good microscope, you can see that not only do the wings have teeny, teeny, teeny hairs, but also their entire body does. And that actually helps them to be, repel water as they're emerging, and helps ensure their success in getting from water to air. But it also imparts a little bit of a dull color to the dun. So we say that duns are dull in color as opposed to spinners, which are shiny. So let me go back to that picture. The mayfly duns, when you look at their wings, they're opaque, very dull in color. You can't see through them. Whereas when you go to the spinner stage, you can see right through the wings. They're almost like little glass windows. The color in general is more intense because they've shed that skin from the dun to the spinner stage, and now they're very shiny in appearance, and they're just brighter out, and the colors are intensified. A couple other things that are interesting in general between males and females. Obviously, males and females have certain reproductive organs that are different, but going from males to females and looking at the eyes, both in the dun and the spinner stage, males have very large eyes, and that's necessary to be able to find the female in the swarm. So depending on the species, they might have really large red eyes or green eyes or orange eyes, and that's all species dependent. But also what's really interesting in the males, going from duns to spinners, their front legs become very, very long, and also the tails become very, very long. And they need those long front legs to be able to grab the females in the swarm and grab her, and they mate in the air. So, and I suspect that the tails then are long also, kind of a secondary sexual characteristic, and also maybe to help balance them in flight with the long front legs. This is a nymph emerging into the dun stage, and you can see that shock being shed. And this happens, fairly quickly. I remember when I first got into fly fishing, and you would read about this, and they would say, oh, it might take a few minutes, but actually it happens pretty quickly. And I have some And it depends on the species and the body size. So a bigger mayfly will take longer. Smaller mayflies are going to emerge fairly quickly. And I have a little video to show you in a bit, and we'll look at some of that emergence. This is the dun molting into the spinner stage, which we don't typically see. This happens away from the river, often under cover of darkness, not always. But that little shock hat, they have to kind of just shed that. It's kind of like just slipping a glove off, and then the adult will emerge. And then hopefully they are able to make it back to the river to mate and reproduce, and then fall to the river so we can catch fish. So remember when I talked about mayfly nymphs, and I said they typically have 3 tails and their gills are on their abdomen. And if you carefully look at each of these, some of the gills are kind of, you know, a little more hard to see. Some are very obvious. They all have 3 tails. And so these are all mayfly nymphs, even though they don't look anything like each other. And all of these different nymphs have different little niches that they occupy. And I'm kind of getting back to the variable that I mentioned early on about habitat. And I think it was, you know, one of the early authors, I think it was Dave Whitlock, termed these different descriptors for each of these mayfly nymphs: clingers, crawlers, swimmers, and burrowers. And as you, you know, spend time on a river and you're turning rocks over or wood over or seining for insects, you will find all of these different mayfly nymphs in different types of habitat. And so I have a program that I talk about where it's just about mayflies, and we go into real depth on where do clingers live and what do the adults look like. And you can make some great analogies as far as, you know, each of these different mayfly nymphs and what the adults look like. So for example, now that I brought it up, the clingers, I don't think my pointer works. The clinger type nymph, all of those adults are Cahill type adults. They all have 2 tails as adults. The burrowers are, you know, the hexes and the white flies and the brown drakes. Those are all our burrowers. Some of those have 2, some of those have 3. The swimmer mayflies, all of those adults also have 2 tails as adults. And the crawlers, as it turns out, all have 3 tails as adults. So some interesting things. I don't want to get too far into that, but I'm going to just pick one of these categories, the crawlers, since I went down this rabbit hole a little bit. So crawlers, a primary example of that are Hendricksons, also sulfurs, and some others. As nymphs, they have to live in a very gravelly type substrate. So, you know, you're going to find a lot of those in Michigan. As those of you that fish maybe up in the Wisconsin driftless area, for example, there are fewer of those there than there are in Michigan because there's not as much gravel in some of the stretches of water up there. But those of you that, well, I'll come back to that. But when you think about these nymphs living in that substrate, they can't just be up and swimming around all the time, or the fish would eat them with abandon. So you really only find the nymph in the water column, meaning from the substrate up to the surface, only right before they're getting ready to emerge, or maybe due to a disturbance like a big spate or flood, or due to drift. And of course, you guys being the DRiFT club, probably, you know, you've done a little research on that. And drift is a natural phenomenon whereby certain insects will just let go of wherever they're living and drift downstream a little bit. This will happen, for example, if there's a new predator that has come into the area, another little, so with a Hendrickson, maybe there's a bigger stonefly nymph that might eat them. And so that's pretty threatening, and maybe that Hendrickson nymph will drift downstream. Other insects will drift maybe if the food patch, if they're feeding on algae or detritus, if it's all gone, they gotta move on. So, but this drift happens at a couple peak times a day. So it happens at dusk and it happens at dawn, and it also happens at 2 a.m., but we're not really often out there at that time of day. But dawn and dusk are prime times for nymphs to be active and moving. So what does that mean fishing wise? So if you fish with a nymph, those are good times of day to fish, especially beadhead especially are effective because you want to keep it down when they drift. It's not like they swim up and take off and go for this nice swim. They're probably only moving a short distance for the most part, maybe only a few inches, maybe sometimes less, sometimes more, but you want to keep your nymph fly down in that substrate and let it drift that way. And again, if it's I like to fish these with a little bit of action. Some nymphs are pretty good swimmers, some are less good at swimming. And so you kind of want to key in on what the natural insect is actually capable of. So crawler type insects, crawler type mayflies, as adults, all have very plain wings and they all have 3 tails. But also interesting in this group, in the crawler group, all the males and females are very unique in that their males are often a different color than females. Kind of like if you're, certain birds, I especially think of cardinals. I mean, there's obviously when you look at a male and female cardinal, the profile is distinctive, but the male's obviously very red and the female's kind of more brownish. You can tell right away that this is a male by looking at the eyes. You can see those great big eyes, and so you know that it's a male. So as a beginner, fly fisher looking at this, I could tell, maybe I couldn't identify it, but I could get an idea of the size, and I could see that it's a dun because it has dull wings. I could tell that it was a male. And then, then I'd have to look at my hatch guide. Here's the female Hendrickson. And so the wings and everything look very similar. I'm going to go back, look at the wings and the venation. And then here's the female. So very similar, but she's just a little bit different color. Instead of that pinky red, she's kind of an olivey browny color. And also in general, the females are a little bit larger, and that's true for almost all insects. The females are a little bit bigger because their abdomens are bigger because they're going to be laying eggs. Here's the male, just a little black and white to show you how similar they look. There's the male and there's the female. And those of you that, have some of the really old literature, imagine looking at insects in black and white in some of the old insect guides. And it was like, well, how are you ever supposed to master any of this with black and white photos? So just as a practical application, does it really matter? I mean, so, I just said that females are bigger, and when you go into a fly shop or you look in your hatch guide, you'll see that there's a range. So for Hendricksons, it will say size 12 to 14. And that doesn't mean use, something about this big. What it really means is the bigger size is the size of the female. The smaller size is the size of the male. So you got males that are smaller in a certain color and females that are a little bit bigger in a different color. So does it really matter? Do fish ever really key in on one or the other? So the answer is sometimes they do. And so biologically, what happens in any population is that the males will hatch out first. And so if you collect a whole bunch of one taxon, if I collect Hendricksons and I hatch them out for 2 or 3 or 4 days, only males will be hatching out. And then pretty soon the females come along and they'll be hatching, and you'll still have males still coming on and females, and then males and females. And so sometimes fish can get keyed in initially on that smaller size. So I think a lot of times when I'm pretty sure that I know that, this is what's hatching, but I'm not catching, I will drop a size fly. First I'll drop a tippet size thinking that that's the issue, but then I'll drop a size in flies, and oftentimes that'll do the trick, and the fish will be keen on the smaller size. After a while, they'll eat both, but sometimes they can get kind of picky. So here's the Hendrickson spinner. And again, you can tell right away it's a male, not because I wrote it there, but the great big eyes, number one. And then look how huge those front legs are and the long, long tails. So here again, as a tying club, especially, we learn that, okay, the tail should be the same length as the body. But in the spinners, certainly in the males, this can be very exaggerated, like they might be at least twice. However, here's a little something to ponder. A lot of times after mating, the males don't necessarily fall to the water. For sure, the females have to come back to the water. They got to lay their eggs, but the males sometimes might just fly away. There's no reason that they have to come back to the water. Some of them will fall there because they're exhausted, and that's that. But so tying wise with spinners, a lot of times we're imitating the female, and oftentimes we'll just incorporate that egg pattern right into the tie. And so we typically still keep the tails about the same length as the body. But generally, proportionally, I mean, if you're counting, there are going to be more females on the water than there will be males. So I like to include that egg sac color right in the pattern. And in my guide, I try to include that information. That egg sac isn't, I mean, in the Hendricksons, it's just kind of bright yellow, but in other things, it does change color. Okay, so here's some very amateur photography or videography. This is me sitting at a tank for hours and days at a time, trying to capture what's happening. So here's the Hendrickson, and this is in one of my tanks, and it's getting active. You can see those gills. You can see it kind of, that scorpion light action is actually a defensive posture. Probably doesn't like me watching it, but it's a good defensive posture, and they've done studies that shown that fish are actually deterred by that and won't eat them when they're doing that, which is kind of interesting.
Hey, could we interrupt for 1 second and just back up slightly? We had a question from Jim Warren. He wanted to know if that Hendrickson egg sac was 1 egg or hundreds of eggs or thousands of eggs.
Well, that's a great question. So that is a big egg cluster, and the way that that is dispensed, and this varies in different species, they will actually, it's like a big golf ball. They'll want to get that whole clump out and into the water. And so they have to kind of, with Hendrickson's, they'll fly up and down and try to, it's kind of a sticky mass, try to get that, out of their bodies into the water. So there are hundreds, if not, in Hendrickson's, probably hundreds of eggs in there. And other insects, or other mayflies, it's a little bit different. But once she dispenses with that big egg ball, then she's all done.
Now, will that egg ball stay together, and does it serve an advantage to that particular species, or will that break up and then just kind of scatter in the water column?
Well, it's kind of a big ball as it goes down, and it's a little bit sticky, and so it will stay down there. And those, in that particular species, those will begin to develop soon after, and then they'll kind of disseminate. Whereas other mayflies, they may lay their eggs, and they'll come out somewhat more individually. But they may overwinter as an egg. Like, for example, in the gray drakes, they get laid and then they don't really start to develop until like January, which is really kind of strange. So there's a little bit of variability with different species.
And then lastly, from a fishing perspective, how vital is that stage as far as having that fly in your fly box, and how long does that stage occur? Hours, an afternoon?
Okay, so great question. So imagine all those hundreds of eggs from one female and extrapolate that out. Not all of them are going to live, and then they, become nymphs, and not all of them are going to live, but a lot of them become duns. Some of those will be eaten by birds and spiders and other insects. And then, still many of that same clutch of eggs will become spinners. That's just from one individual. But with any one species, you will have, upwards of a couple, 2 weeks, maybe 3 weeks in any one location. Maybe if you just stayed in one location, maybe a couple of weeks. It's hard to say, maybe 2 to 3 weeks. I'm just going to be general there. You will have those spinners then for many nights, depending on, because you've got so many different aged eggs all within a couple of weeks. So even though they're mass emergences, it's not as though they all come out on the same day at the same time. So there is a little window in there. So you'll want that spinner in your fly box. And that's another great thing. That'd be a really fun talk to have, and how to set up your fly box. I'm getting a little bit away from my talk, but I mean, the way I set up my fly boxes is completely by season. I have my panfish boxes, but my trout boxes are totally set up by season. So I've got early season, Hendrickson's, sulfurs, blue-winged olives, mahoganies. And then I have a little window of transition. Then I get into brown drakes, hex, and the bigger ones, isonychias. And then you transition into trichos and things, and then terrestrials. So, you can't carry every possible fly, so that, it takes you half an hour to choose a fly. You got to be confident in saying, I'm going to this region. I know the phenology is this order. And so I'm confidently carrying these flies in my fly box. So, yeah, I hope that answers your question.
And you feel that a trout will key in on that egg sac as a fly?
On a fly, yeah, for sure. I mean, I add that if it gives it a color point, number one. And fish are often interested in that. But if you see those spinners on the water, when you see them in flight, even, I mean, it looks almost like a little lantern coming along. And with Hendrickson in particular, those everything in that in this crawler group, they actually have that, they kind of exude that little ball right before they're coming down to the water. So I think especially with this group, an egg sac is really critical. Now, if you go to something like brown drakes or hexes, the way they lay their eggs, they actually they come out more tubular. And so they hit the water and squeeze like a big double-barreled toothpaste tube, for lack of, that's a pretty good descriptor, actually. So not everything that hits the water has that egg sac ready to go. Many of the caddis do, for example. Many of the stoneflies do. And so if you know it, I put that little egg sac right on the spinner stage.
Good, thank you. I'm sorry for interrupting.
No, those are great, great questions, and I appreciate that. So we saw that one nymph just kind of acting defensively. Here's a couple nymphs. They're interacting. It doesn't really mean anything, but this shows them starting to become active and kind of falling back down to the ground. So right before an emergence, nymphs will become more active. And so when, you go somewhere, I know most of you guys are in Illinois, so I know you're traveling to fish. So you're either going to Wisconsin or Michigan for the most part, maybe to Iowa. But in fishing a nymph, that sort of activity is starting to happen. So before you see any insects on the water, that's a good time to use a nymph and know that they're getting active right before the duns get ready to hatch. Here's a nymph that has swam up to the surface. Some of you have read Kelly Gallup's books, and he coined a term, cripples and stillborns. And some mayfly nymphs will never open. They just sort of get stuck here. And for whatever reason, the thorax won't split open and the nymphs, or the wings don't come out of the nymph. And they're really pretty much sitting ducks for fish. And a lot of times fish will feed on these, and there's no way to know what percentage are eating them up here versus down at the bottom. But it's basically a dud, and fish selectively can feed on them. So once in a while, things work out, and the nymph will swim to the surface. And then quickly emerge, and it's ready to go then. And you can see how light in color that's a female there. But a lot of these, as the dun hatches, the color will be light, and after it flies away and we see it in, later on, the color will get a little bit more intense. Here's looking down, and you can tell this is a male with the big eyes, but there's a dun that just emerged. And look to your lower left-hand corner, and here's another nymph, and boom, just like that, that takes off and it's ready to go. So just watch that one more time. So So when you're on the water, a lot of times you'll have hundreds, if not more, of this going on at once. But kind of the takeaway of this is, when you read the literature, it'll say, oh, it takes such and such a time to hatch, and the insects are on the water for this long. But really, they're not. They get up and out of there quickly, and they have to, or fish would just be having a feeding frenzy, and none of them would make it. But the second part of that is you have to be able to tie your knots really fast. If you're changing out flies, you got to know what flies to anticipate. Early in the season, it's not that challenging. There's only a couple different hatches early on, but as you get into further into the season, mid-season, that sometimes there might be 5 or 6 different bugs going, and you got to know what's happening, and you got to be able to change flies really quickly in order to take part in that and have success. So we saw something that was called a stillborn. If you look to the left, that is what Kelly called a cripple. Here it is up close, and that it got up and it got on top of the water, but for whatever reason, those wings are not ever going to open up any further. And if you look right there, you can see the left-hand rear wing has almost made it out. It's not quite filled out with fluid, but the other wings are never going to make it. And I'm sure all of you have been in situations where you like, oh, I know this is what the bug is, but they're not eating any of my flies. And sometimes these will get into little back eddies, and fish will just sit there and pick those cripples off. And you can tie a cripple, but you can also just take a pattern that you already have in your box and like cut half of the hackle off, and so that it falls over and looks a little bit deformed, and use that as a cripple pattern. So let's look at, just kind of think through some practical applications of what we just saw there in the video. And when you're on the water, think about what life cycle stage the fish are feeding on: nymph, emerger, dun, spinner. Maybe they're eating one of those other weird things, a stillborn or cripple. So look at the fish rise. And if you're on the water and you just see a bulge, no fish part comes up, but something just oof like that, then it's eating something right under the water. It's got to be a nymph or an emerger, or possibly a stillborn. That's a little bit more closer to the surface, but a bulge is definitely typically a nymph or an emerger. If you see that the fish body, like dorsal fin and tail, come up, so it's feeding a little bit like this, it's eating an emerger. If you see body come up and porpoise on it, it's typically emerger or dun. And then honestly, we didn't see this in the video, but the easiest fish rise to read is the spinner, where all you ever see is just the mouth, just a delicate thing. There's no real immediacy to it, because they know the spinners aren't going anywhere and are just dying. So go back to the beginning now, when I brought up all those variables, and think about the Hendrickson hatch. So geographically, it only occurs in the eastern United States. You're not going to find it if you go out west. Phenologically, it occurs around opening day, so right at the end of April into mid-May. Sizes are going to be a size 12 to 14, with the larger size being the female, the smaller being the male. Time of day, the duns are going to hatch in mid to late afternoon, and then you're going to have to come back early evening for the spinners. This year, we don't have to worry about it getting too hot, but some years if you have a really warm spring, sometimes they won't actually have they won't actually spin until morning if it gets really hot. But probably the most important thing stream wise is you've got to find a section of water that has gravelly habitat so that you actually encounter Hendricksons at all. Here's just a little commercial. This is a nifty little tool here called a quick seine. And some of you might have this. It's a little Velcro, it Velcros around your net. And it's just a little bit of mesh and that will unfold and go over your net. I used to just carry this kind of netting and then try to hold it in the water column, right? Half in and half out. And then I could collect insects and say, oh yeah, this is what's on the water. And this is a nifty little device that goes right over your net. Even if you can't identify them, if maybe you're not at that, proficiency, you can still at least say, oh, hopefully you can say it's a mayfly or a stonefly or a caddisfly or something else, maybe, greenfly or midge or something else. But you can also at least say one of those items, and then you can say, well, it's about this size, and gee, I have this fly in my fly box, and I can make a pretty good match with that, and then tie it on and imitate it that way. Okay, any other mayfly questions? I'm going to talk now a little bit about stoneflies. So with each of those different groups of the, crawlers, swimmers, clingers, and burrowers, you can kind of go down a rabbit hole with each of those and talk about behavior and habitat, and it's pretty fun and interesting, at least for me. Okay, well, if no questions, and you can always ask me at the end too if you think of something. So let's kind of segue into stoneflies. Here is another group with an incomplete metamorphosis, but it's not quite as complicated. The female there on the left will lay her eggs, and they will grow up and develop mostly over the course of a year. We do have some that really in further northern regions that take upwards of 2 to 3 years, but most everything in our region takes about a year to develop. And they'll grow and slowly change. And then after about a year, instead of swimming to the surface, they're going to crawl out to a stone or a rock or maybe the bank or a big dead log. And they're going to just crawl out and emerge. And then instead of having a big mating fest like the mayflies did, they actually often will mate immediately after they emerge. Sometimes right there, if they're males and females, males will emerge first again. And so then as the females come out, they immediately find the females and they'll mate. And then they'll fly off. They do have mouth parts, and they'll live for sometimes a couple of weeks. And again, the males aren't going to come back to the river. So after, all is said and done, it's just the females will come back and lay their eggs. And once those eggs are laid, then she might fall in the river and die, or if she's lucky, she lays all of her eggs and she might fly off, and she's still going to die. I mean, they just don't live that long. If you're fishing out west, because they have they have a lot more stoneflies out there. Sometimes you'll have whole stream sides where the adults will be on the leaves and things, and they've crawled up and they're just still kind of hanging there after they've emerged. We don't really have that kind of emergence here in the Midwest, but we do have a number of species of stoneflies. But as anglers, we're really only imitating the female laying her eggs and the nymphs in the water. A stonefly nymph, you can tell very readily it has 2 tails and 2 long antennae, 2 sets of wing pads. Some of them are, have kind of ornamented, wing pads and thorax regions and head regions. Not all of them, but basically 2, 2, and 2 on body parts. Some stoneflies have various gill tufts under their legs, some under their chin, some down in the tail region. Some don't have any at all, and they really take in most of their oxygen right through their exoskeleton. And so in saying that, stoneflies have to live in very cold, very well oxygenated water for that reason. And so out west, because they have a lot of mountainous runoff, they have a lot more stoneflies than we do. Very highly oxygenated water there with the turbidity, or just, a lot of runoff. So a stonefly adult kind of looks cockroachy, but much more attractive. They have 2 pair of wings that are completely folded and held over their backs. They have 2 pretty long antenna, longish antenna. Sometimes they have 2 tails. Sometimes in the adult stage, you don't see the tails at all. They're not the best flyers. In fact, they spend as much time scampering around and crawling quickly as they do in flight until, of course, the female has to come back and lay her eggs. This is what it looks like when the nymph crawls up on a log and splits along the thorax and starts to emerge. Again, a little bit like that one mayfly, the Hendrickson we watched emerge, how light she was. You can see how very pale the color is when they first emerge. This often is under cover of darkness, especially once you're into summertime. And it's important that it happens then because it's a very long process. It takes about 45 minutes. For some of the smaller stoneflies, I've watched them emerge and photograph them, and they might take 30 minutes, but still it's a long time. So you can imagine mice and other mammals crawling around at night, just feeding on these as they emerge. And so if they emerged in the daytime, they'd really be in trouble. So we don't typically see it. Usually we just see the shucks on the rocks the next day and we say, oh, stoneflies emerged. So in fishing and stonefly nymphs, you got to think about what I just said there. So they crawl, the stoneflies crawl out to emerge. So if you're fishing a stonefly nymph right before, say you see some shucks on the side, on the rocks and such, that's a good time then to say, oh, well, stoneflies are beginning to emerge. I think I'm going to fish a stonefly nymph. And this I can see from the shuck about what size it was, and I might even be able to identify it. But what you don't want to do is stand on the side of the river and cast to the middle. What you want to do is get out into the middle and then cast back to the side, because as a population, what's happening before an emergence is those stonefly nymphs are all crawling out to the banks and the fish are following them. And so the fish will be keyed in on that movement. And that's what you want to do with your fly. Now you'll see that I put a little white pattern in there and you probably think, well, that's weird. What's that all about? But right, and this is true with mayflies as well. When they go to molt, the nymphs molt and change. And every time they have to, shed that skin, they're completely white. afterwards, and they'll stay that way for quite a long time, upwards of a half day or so. And so you can actually fish a little white pattern, and it's kind of fun to try to do this. They typically are molting right at dusk or dawn, right at that drift time. And so that's a good time if you want to just kind of do it for fun and see if you catch anything, that would be the time to imitate one of those.
So quick question, Anne, when you're tying patterns of dry flies or imitating a particular fly that you want to fish, what is more important to you, the size or the color?
That's a great question. So for me, the size is most important, and profile and color is the last thing on my list. And in really some of the old, I don't know, tying things. So some of the old literature, the color was a big focus, but sometimes, unless you're in incredibly still water, and fish can key in on all those different variables, I think color is oftentimes, we as Tyers get fixated on that, and we buy inordinate numbers of different products. And I really think for me, I key in on size and profile. And as far as emergers, I have gotten away from parachutes and keyed in on more almost half in, half out of the water type emerger patterns, and find them to be more successful.
Wow, okay. Now,
I'm not saying don't give up on parachutes, but for me, stuff with CDC as an emerger, wing, or snowshoe hair, I like those materials better. I just, anything where you're half in the water and half out when you're imitating the emerger done stages is I think better.
Okay. So you're out fishing, you have a handful of boxes, you have your springtime mixture. If color's not that hugely important to you, do you have a tendency to kind of tie stuff in a certain hue that you feel works for you, keeping in mind that you have simulated the profile and the size?
No, so I might have misspoke there. So, like for isonychias, I know that color, and I'm still going to have that color. I'm still going to have my isonychia patterns, still going to have my Hendrickson patterns. I'm still keying in on specific taxon, but if somebody says you got to have this hue of dubbing, I'm not going to buy any more packages of dubbing. And the other thing is, let me just throw this into the mix. In that crawler group of mayflies, and that's the one I walked you through with the Hendricksons. Not only do you have different color male and female, but you can go to different river systems with little tweaks in water chemistry, and you'll get different body tints. So
just like fish.
Yeah, exactly. So I think fish are more forgiving of color than we are as fly tyers.
Okay.
So I say get your body size right, get your profile right, and then do the right color, but don't go overboard on that, and don't feel. I've only in one case ever had a fish where I had to totally have the right color, and it was a hopper. It was like they were eating only orange hoppers. I was out west, and I had yellow, I had green, I had this, and it was like finally like one orange hopper. It's the only time in my whole life where I ever had to have the exact right color. So,
Experience it, and my guide had 4 different Sharpies in his boat. So if he didn't have, he would change the bottom to yellow. He would get them in tan and then change it to whatever the color of the bottom needed to be.
with hoppers.
Yeah,
Yeah.
yeah.
That's the only time I've ever been like, seriously, are you serious? So, everything else has, it's like, I just think as Tyers, sometimes we get a little too
carried away.
Yeah, a little bit. Yeah. I mean, you know the right color, but don't overthink it.
Okay, thank you.
You're welcome. So here's back to stoneflies. So here's the underside of a stonefly, and you can see that a lot of, so let's go back to that Hendrickson female where she had like the golf ball egg, and she was going to dispel that whole cluster at once. With the stoneflies, they're more, you can see the individual eggs, and every time she hits the water, she's going to dispel a clump, and then she's going to fly back up and then squeeze out more. So you can see how her abdomen has shrunk a lot. I mean, it's probably half the size of once it was, and it's like like a toothpaste tube, and she'll try to get more and more of those out of there. And in case you're thinking of it, with a lot of insects, the eggs can pass through a fish's alimentary canal and still be intact. So even if she gets eaten, a lot of these eggs will still be viable and be able to hatch after they're not entirely, they go through so fast, they're not really digested. So in fishing stonefly patterns, the female is never really sitting on the water, just hanging out. She's in and out and in and out and dipping her abdomen. So you really want to be moving that pattern around and picking it up and down. And a lot of times when fish will eat it is when you just go to lift it up and it's watching it. And then as you lift it, that's when you'll get your take, which makes it a little bit challenging because a lot of times you miss it because you're just picking it up. Okay, last group we'll go through and then we'll have some questions. Is the caddis. And here's a case of the first group of insects that were complete metamorphosis. And so we actually have like a chrysalis or pupa stage here. So this is a little bit equivalent to our monarch butterfly, the one life cycle that we all grew up with. So the female will lay her eggs, and they will grow into these little grub-like larvae, little maggoty things. And in case you wondered, when you go to a fly shop, you got larval flies and you got nymph flies. The nymphs are all the incomplete insects, incomplete metamorphosis, caddis and mayflies and such. And then the things that are larvae are things with complete metamorphosis, things like caddis, and just little trivia thing there for you. So with caddis, they're kind of an evolutionary, more advanced group of insects. So some caddis larvae are free living and will move around on its substrate freely and just graze and sometimes even eat other small little insects. Other caddis are very interesting in that they build a case, sometimes out of little bits of stone or wood, plant material, bits of gravel, bits of shell. Other caddis larvae actually will spin a little web. Some of those, they'll live in the whole little, like a web sack. Others build like a little kind of a double-sided cave, and they'll have a a little retreat for themselves. And then over here, they'll have a little web and they'll catch their food there. And then they'll come out of their cave and come over and feed on the food that's collected in the web and then go back in their retreat. So quite an interesting insect. After about a year, they'll build a little pupal retreat. In about 2.5 weeks, they will emerge and swim to the surface and then split along the thorax and hatch and fly off. They will often mate right on the shrubbery. A lot of times you'll be fishing and you'll be kind of waiting, maybe come to a deep hole and you put your hand on an alder or something and you'll just see all these thousands of caddis in there, all mating right in front of you. And then again, they can live for a couple of weeks and then the females will come back and lay their eggs. They can do that either by dipping their abdomens, but many of them actually dive down into the water and swim down and lay their eggs on rocks. So as anglers, we can imitate all these different stages of the life cycle. Quite often, some of the more successful stages are the emerging pupa. Once the caddis are such good flyers that a lot of times, you'll see thousands of them, right above the water surface. You never see any fish feeding on them, but some will feed on the female laying her eggs. But oftentimes we have good success, especially in tailwater situations. We have a lot of species of caddis that lay their eggs underwater, and then you can use a diving caddis for that.
And with that being said, why is the elk-hair caddis such a prolific fly pattern?
I'm going to come back to that in just a minute. So here's a larva pattern that's the free living. Many of these are kind of bright in color and live in typically more highly oxygenated waters. But they're evolutionarily, they're more primitive of the caddis, and of course easy to imitate with any little grub-like pattern. Here's a case caddis, and with any of the case caddis, you can oftentimes identify right down to the genus what it is just based on the case materials. If you look at the lower right, you can see the little legs sticking out, but you can't see the insect in there at all. You would think that would be great camouflage, and it is, but it's odd that fish do eventually see these moving around, and they do. Some of the older fish will learn that they can eat these and that there's protein in there. Different times of the year when these become very active, fish can and do feed on them. Here's I put this in as just to show you an assortment of different case materials. You can look at each of these and see little bits of plant material, gravel, stone, sand, shells. The caddis up in the upper right is actually one of those that builds the little web retreat and kind of lives in one side and then just sticks its head out and feeds on in the little web that it's built to collect its material. Here is a caddis pupa that's just, a very short amount of time away from emerging. And I like to equate this to our everyday plastic material where it's all you can do to get the item that you're purchasing out of the plastic. And all of this packaging for the pupa ensures that it can get to the water surface without, efficiently and without getting any of the body parts wet. And it's aerodynamic or water dynamic. I don't even know what that word is, but gets to the surface quickly you can see the wings there. They're just totally packed in there, just like a little parachute, the long antenna, and it's ready to go. And then the caddis adult itself is, sometimes they're not very, fairly drab in appearance, too long antenna, no tails, and they hold their wings in a tent-like fashion. Again, as Tyers, we always tie the wings to the tip of the abdomen with caddis, even though the wings are always at least another third as long as the body itself. Second here, many of our caddis, so we always tie the elk-hair caddis with, kind of a tan body, but as Tyers, you should always have plenty of green-bodied caddis, because a lot of caddis bodies are green. This is a tailwater species, a Chematopsyche, but that vivid green body there will actually darken, and that's true, well, with all of our insects, but especially with caddis. So it might be bright green when it emerges. So those of you that are thinking, oh, I should tie this. So if you're tying emergent patterns, and then you have other patterns that you're going to fish in that life cycle, just know that the bodies will be a little bit darker in the adult stage. These are all caddis, so they're not all brown and boring like we tend to think of caddis, especially like the top right. Some have very highly ornamented bodies. Some have, in the lower left, the antenna on that insect extend at least another 1/3, and so they're kind of an interesting group, but a challenge to identify more so than some of the other groups, but really fascinating. In fishing patterns, though, many of the caddis drift, and so I've got some. This slide shows some cased patterns. And you can use a case pattern to imitate an insect. It can be fun, and fish do eat them. It's probably not, the choice way to target fish, but it can be really fun. But with my favorite is fishing the emerging pupa, and this particular pattern I think is really amazing. This tyer, some of you might have known Oscar Filu, but he was really quite a fine tyer and tied some beautiful patterns. But you can see that perfect, like the real life one I just showed you. You can see how well he's done in imitating that. They're excellent swimmers, so I like to fish these, on the swing, and as soon as it gets to the swing ends and it starts to rise, that's typically when fish will eat those. So in answer to your question there, Mark, so why is the elk-hair caddis so popular? So I probably should know this, but I don't really know where that pattern originated, but I think it's a great pattern for some kinds of water. So if you have water with good choppy current on it, the elk-hair caddis is probably great. But this top left pattern, that's similar to an elk-hair, but without all of the hackle in it. That's actually called a slow water caddis. It's going to sit more in the film. I've been in situations, well, this actually was out west, but you can get this in any tailwater. So it was on the Missouri River, but in Michigan, our tailwaters aren't really like tailwaters anymore. I'll come back to that. But in those kind of situations, the caddis are sitting in very slow water, and you've got to have, you can't use an elk-hair caddis there and have any success. They want a pattern that sits right in the film. So sometimes, we would actually have to cut the hackle off the bottom So it would sit in the film or go back later in the day and tie some CDC patterns that really were directly in the film. Personally, my favorite is an X caddis for any kind of emergence, and I'll tie X caddis in a number of sizes with different body colors, and I find that to be, you know, a really good successful pattern. So I don't tie elk hair caddis anymore. It's a pretty pattern. It's going to be good in certain situations, but I tie X caddis, I tie CDC caddis, and I'll use an elk hair caddis if I'm in water with pretty good current and pretty good chop on it. But that's my story, and I'm sticking to it.
You said you've gotten away from parachutes.
Well, parachutes are more of that's a mayfly indicator or pattern. I still have them, but I have gotten into a parachute is really designed to imitate the done stage, and you can tailor that to actually, I mean, you can cut out parts of the parachute and turn it into a spinner pattern. But I just find emerger patterns where you're half in the water and half out to be just a lot more successful.
Like a clink hammer or something?
Yep, exactly.
Okay.
Love that style, whether you're fishing the clink hammer with the hook or you're modifying it in some way. So in the end, I hope I didn't go too long here, but what I want you to, the takeaway is to stop relying on your fishing partner and make some of those decisions yourself. And in the end, I hope that not only will your fly fishing be better, but hopefully your fish catching will be better in the end. So again, I'm going to put my email up there, and I'm working on this second edition. I won't have the same cover. I might have to modify the title a little bit, but I'll let the clubs know when this is ready. But if you send me an email, I've got a long list going, and I'll let you know when it comes out. And I'm going to be I haven't thought all this through, but I think right now I'm going to try to manage the distributorship as well. And of course it'll be in fly shops okay. So any questions? I know we've had a lot, and maybe.
Yeah,
I had a question.
there you go.
Yeah, I had a question about the pupal stage of the caddisfly. And I know, like you were doing too, I compare it with the chrysalis stage of a butterfly. And I guess what I'm trying to say is, when it's in the pupal stage, is it still active? Does it still eat? Does it still swim?
Okay, that's a great question. So when it's in a pupal stage, imagine that it's in an enclosure of some kind. So,
right.
So for some of the case caddis, they will just seal up most of the end of the case. And then to answer your question, yes, it's active in there as far as radically changing. It's not eating, but if you think of a big caterpillar, a big grub, it has to completely transform and metamorphosize in there. So it's active in that sense, but when it's completely in that pupation stage, it's largely unavailable to fish. I mean, they're not going to be able to find it. It's not moving around in there. It's going to be stationary. And so it's really only available as it emerges.
I see. So I'm this is kind of a different follow-up on when you were talking about the clink hammer versus a parachute, because I fish a lot of clink hammers also. And that was one thing I had noticed too, was that the fish seem like that body that's hanging down as an emerger. And then the other thing I noticed too was that I fish soft tackles a lot, and a lot of times I fish a dead drift. and the fishing treat that as an emerger also, because usually it's maybe like an inch or two below the surface. And I get a lot of stress that one.
Yes. And I fish a lot of soft tackles, and there's a place where you can, play with materials and,
Mhm.
incorporate peacock. There's or CDC. I love CDC, and there's properties in there that are very live-like. But again, I think a lot of it is, that subsurface stuff, the whole rise, that whole technique is what makes it super successful. And for me, in all these years of fishing and just fishing that, going back to mayflies now, where you're fishing a perfectly still parachute, and after I've watched a lot of insects emerge, I think about trout, and then they got time to come up. Look at that. I'm not saying that I never use them, but I'm just saying I've really graduated away from those to something that's just more lifelike, hanging down, and where I might fish it with a little more activity, and not, I'm not afraid to move a fly. I mean, when in the little bit of video that we looked at, you can see how much activity is going on. And when I was first introduced to fly fishing, it's like, oh, men, you do want a nice presentation, but if they're not going to touch that fly, it's like, I'm going to try moving it. I'm going to experiment. And I've had, good success in just thinking a little bit more out of the box.
Yeah.
Especially with caddis
Right.
always moving, stoneflies always moving.
Mhm.
So
That's true. Because I noticed that too. And the one thing I come across was I noticed that the lower my dry flies rode in the current, the better the hookup seemed to get, and the more solid strikes I seem to get. And I did try to activate mine a couple of times, and it did work. But after listening to you, I think I've got to try that more.
yeah. And so for me, when I think about the one time I am being a little less active with my insect is with mayfly spinners, because they're exhausted, they're on the water, they really aren't moving. And if you take the spinner stage of the mayfly, once those wings get down in the water, they're stuck. It's really hard for them to get out of the water. And remember when I showed you the picture of the dun with tiny little bitty hairs that you could see, that actually helps them get up and out of the water. Whereas with the spinner, they don't have any of that. So once they're down, it's like a wet Kleenex on the water. It's really challenging for them to get up and out of there again. They're just, surface tension sticks them down. So fishing wise, men with spinners, and really do keep them nice and still, because the real life insect is dying and not really moving around.
All right. Okay. Thank you.
You're welcome. Anybody else?
Yeah, and I've got a question. Thinking back to that video of the mayfly nymph arching its back into the scorpion type position, and you mentioned that would be a deterrent to fish. That makes me think I've been tying my jig hooks for no point. I mean, if I'm tying a mayfly on a beadhead jig hook so the hook rides hook up, that would be mimicking that behavior.
A little bit, for sure.
Okay.
So that's just one. That was like the crawler group.
Sure.
And I'm working on trying to get a lot more video of all these different groups so that you can see them in action. And like, if you look at the swimmer group, that's entirely different. So things like blue-winged olive, isonychias, they're not going to be behaving the same way at all. And it's just fun to watch all of those. but anyway, it's just one little glimpse. And I'm honestly to get anything at all. I mean, literally, and it's not even that good a video, but it does show you something. But I'll spend days and days
Oh,
and days.
It's amazing to see that behavior.
It's so fun. It's just, I just love it. I just am like, oh, that's so neat, and how can I apply that then? And I've got some video of hex hatching, and they're so much different. The hexagenia, they're much bigger. And so it takes them, so those Hendricksons were hatching in seconds, and the hexagenia, it might take them about a minute. So it's got to be, just different strategy for them to be able to survive. And I don't know. I just spend a lot of time thinking about all this and extrapolating.
I think the thing that impresses me most, Anne, about your aquatic containers, being a person that had tropical fish for many years, is your substrate looks so natural. That had to be a lot of work to create that.
It was, yeah. And so it was, yeah. So and of course, a lot of it was trial and error, just like, okay, that didn't work. Let's think of something else. Let's try something else. And so,
So how-
and hauling all of that river water back home and not having my husband leave me or anything.
you would come home with containers of Rubbermaids with rocks and moss and all kinds of stuff in it. And just
I did, and gallons and gallons and gallons of water, and then all these different containers of bugs. And it's like,
wow,
you know,
that would have made a good movie in itself.
I wish I had taken more pictures along the way, because, well, here's a funny story. When I was doing this, my kids, the older two were in high school, and the younger one was like late elementary or something. And and so I'm also trying to cart them to dance and pick them up from school and go to football games and all this. And before I had really figured out how to create a scenario to photograph the adults, I was losing a lot of bugs, and I'd chase them around this room. And so if you had something that you really needed, it was kind of like baseball cards, and it's like, oh, I got a Mickey Mantle, I can't, it's the only one I have. And so I would lose a bug, and then I'd have to go pick them up. I'd pick up the kids, so I'd shut the doors and I'd turn the lights off, and I knew they would fly up. And I remember one time putting a note on the door that says, "Don't go in the basement, bug loose." So the girls read the note on the door, and then literally one of them was driving then. They got back in her car and drove around for 2.5 hours until I got home, until the bug had been captured. So clearly I didn't do the right thing in raising my kids right and loving bugs, but it made me laugh. And it's like, girls, how many times have I told you mayflies don't have mouth parts? Oh,
Well, does anybody else have another question for Anne?
Well, there was a question posted about, and I heard you say it in the first part of your talk, but what is going to be different between the first and the second edition of your book?
so in the second edition, I am going to add terrestrial insects. So I will have a section on crickets, grasshoppers. I'm going to add adult crane flies. I will have a section on cicadas. I will have midge, larva and pupa, which I didn't include in the first. Oh, what else? Beetles, moths. Um, there's a couple more. Deer flies.
Isn't gonna fit in my pocket anymore.
No, I'm being careful. I gotta keep the size the same because that's important, you know, it does fit in your pocket and that's nice. Although I am entertaining just slightly larger so that I can increase the font size. So it'd be easier to read without, you know, huge
And what you ought to do
glasses.
is become the new Ernie Schweibert and come out with that big 2-volume set.
Oh, it's a great reference, but it's a challenge to read.
It certainly is.
Oh, boy. So no, we'll let Ernie be Ernie. I
And
want to be a little more hands-on.
Yeah, I'd almost be afraid that when he was around to ask him a question, because man, you get it. Both of them girls.
No.
And I
Yes.
want to thank you so much for this presentation and telling us about not so much the different stages, but how to fish them too. That was really interesting. Thank you.
Thank you. Thank you. Well, I think, you know, just the more you study them and the more you're willing to make choices and decisions on your own, and learn from that, which, you know, a lot of that I did. And, you know, I think when you first get started, you're very, you know, like I have to do it this way, but like anything else that we learn from our mistakes, and sometimes that's good. I mean, most of the time it's good, because sometimes really good end results come of that.
Hi, Ann. It's Donna Smith.
Hi, Donna.
How are you?
I'm good. How are you?
Doing well. I don't really have a question. I just wanted to say hi.
Well, hi. Thanks for joining in, and I miss seeing you.
I miss seeing you too. PJ and I are doing a school here at our house this weekend. So I'm going to be using some of my notes to talk to the girls about bugs this weekend.
Oh, great. That sounds awesome, Donna.
So thanks very much for joining us today.
Okay, you're welcome.
Well, if nobody has any more questions for Ann, I think we got more than our money's worth. Ann, thank you so very much for your generous amount of time you've spent with us tonight. And I probably speak for most of the members when I say we're looking forward to the second coming of your book. Perhaps what you could do is just send the club or send me a little tickler. I'll send something out and see if we can just get a giant order together or something like that.
Okay, that sounds great.
Make it easier for you. Instead of shipping out 10 or 15 or 20 books, you could just send one package or,
Okay, that'll be great.
okay. Somebody wanted to know, have you established a retail price for it yet or no?
It'll probably be close to the same. I think it was $29.95, and
Okay.
it's an expensive endeavor. The publisher had, in just details, sent it to Singapore or Hong Kong before. I think it was Hong Kong, so they could print it at a very low price, and then, but it was a slow boat to China back home to get them. And literally that publisher went out of business with all the unrest in Hong Kong. Now, why they didn't, secure another printer, I don't know. I think there are issues there, but what do I know? I'm just going to take it in my own hands, and then I don't have to deal with that again.
Yeah.
Well, good luck with that endeavor. It will be an interesting experience, I'm sure.
I'm sure, yes.
Very good. Well, Ann, thank you so very much for joining us tonight. We appreciate your time. It was a very informative presentation, and we look forward to seeing you again in a not too distant future.
Okay, real good. Thanks, everyone. Good to see you
Well,
all.
thank you.
All right,
Thank
thanks,
you.
Ann.
Thank you. It was
All right.
great.
We'll see
Thank
later.
you.
Okay, thank you.
Thanks.
Thank you, Ann.
All right.
Thank
Bye-bye.
you.